Normally, whenever I give a talk in front of my PhD advisor, Mike Eisen, I like to take the opportunity to poke some fun at him. But in this instance, I decided not to after seeing the very public humiliation that he doled out to my postdoc advisor, Eric Lander. So I think I'm just going to let that slide this time in the hopes that I stay on Mike's good side. So today I'd like to tell you about some of the latest unpublished work to come out of my lab. And so one of the main drivers of research in my lab is this classic question of how does genotype affect phenotype? And so the main focus traditionally in this area has been on highly penetrant Mendelian traits that are caused by single protein coding changes of really major effect. And so a very classic example of this would be sickle cell anemia, which is caused by a single amino acid change. Now, in the past 10 years, our ability to associate genotype with phenotype has been transformed by genome-wide association studies, or GWAS. And in these studies, you take thousands of different humans, and you classify them into cases who have a certain trait and controls who don't. And then you genotype each of these individuals at a few million SNPs around the genome, and then you compare the allele frequencies between these two groups. And if you find a significant difference, then you've identified a region of the genome that contributes to the variation in that trait. And so I think one of the most interesting things to come out of the hundreds of GWAS that have been published in the last 10 years is just asking where are these variants that are affecting complex traits located. And in contrast to these Mendelian traits where the main focus has been on the protein coding regions, it's actually pretty striking that only about 10% of the GWAS hits for these more complex traits have fallen into these protein coding regions. Now, for this 10%, it's very easy to immediately say, what is this sequence change doing to the protein sequence? Because, of course, we have the genetic code, so we can just look it up. Now, in contrast, for the 90% of changes, the vast majority that are in these non-coding regions, we cannot do anything like this because we don't have the equivalent of a genetic code for non-coding regions. So really, what's happening in terms of how these genetic variants in the non-coding regions are affecting these traits or gene expression regulation is typically a big mystery. We just don't know. Even though we can identify that they're having an effect on traits, we don't know how they're doing it. And this predominance of non-coding variation is not just seen in GWAS, it's also been seen in studies from my lab as well as David Einard and Dmitry Petrov's lab in recent human adaptations as well with a similar type of percentage. So the question that I want to address today is how is this non-coding genetic variation impacting complex traits? And in particular, at the molecular level, what are these non-coding variants actually doing? And this work that I'm going to tell you about is almost entirely done by one very talented graduate student, Ashley Taranci. So we know quite a bit about transcriptional cis-regulation and in particular the importance of transcription factor binding to elements like enhancers and promoters. And so one potential way that we might be able to understand the molecular mechanism of this non-coding variation would be if we could identify genetic variants such as SNPs that are affecting transcription factor binding. And if we could do that, you could imagine taking a simple Venn diagram like this and saying, on the one hand, we have SNPs that affect transcription factor binding, and on the other, we have SNPs that affect these complex traits. And if we look at the intersection of those, then those would be SNPs that are affecting these traits quite possibly via their effect on transcription factor binding. And so this would be a very easy way to get at this. So then the question is, how could we identify these SNPs affecting TF binding in a genome-wide systematic way? So there's a very widely used method for looking at where a certain protein is binding in the genome called Chromatin immunoprecipitation, or ChIP. And so this lets you isolate the genomic regions where any protein is bound. And then if you sequence those regions, that's known as ChIP-seq. So ChIP-seq has been used by many, many labs, and it can reveal where a TF is binding throughout the genome. But that gives us the binding sites, not the genetic variants that are affecting binding, which is what we really want. So in order to find those genetic variants, one way that you could do that would be to just do this ChIP-seq in many, many different human samples. So if you imagine you have a large population of humans, then you do a ChIP for your factor of interest on each one, and then a separate sequencing of each one, and then you could then correlate the genotypes of these individuals with the binding at each binding site, and you'd be able to map genetic variants affecting binding that way. Now, this has been carried out only on a rather small scale with these individual samples because this is really quite a daunting experiment, both in terms of time and money to carry out. So what we've done in our lab is to come up with a more streamlined version that lets us get at the same question of genetic variants affecting TF binding, but in a much more efficient way. And so the way that we do this is very simple. We just take the same population of humans that you would have done all these separate ChIP-seqs on, but instead of doing all these ones separately, we just take all those individuals and pool them into a single test tube before doing the ChIP. And then we do a single ChIP-seq on that pooled population. Now, how does this actually work? So let's imagine a particular allele with a particular SNP with two alleles. We'll call it red and blue. And then we have a transcription factor, which is this green dot, which binds to some of these alleles. And so the blue allele starts out as the minor allele. It's more rare, but it also has a higher affinity for this transcription factor. So after we do the ChIP, then we see that the allele frequency of the blue allele has actually increased because it had this higher affinity. And so we see a shift in the allele frequency comparing the pre-ChIP and the post-ChIP samples. And this shift in allele frequency is something we would only expect to see if we have a cis-acting genetic variant affecting the binding of this TF. If there were variants not affecting it, then there would be no shift in their allele frequencies. So that's the entire approach right there. We just look for these shifts in allele frequencies. So the two main advantages of this approach is, first of all, it reduces the experimental variability because doing all these ChIP-seqs, there's a lot of variability from batch to batch. But here, since we're just doing one, we don't have to worry about that. And the other advantage is that it gives you a major decrease in the amount of effort and expense compared to this non-pooled brute force approach. So for a pilot experiment, we started out with just a small population of human lymphoblastoid cell lines from Nigeria. And we did ChIP-seq for 5 different transcription factors, as well as a histone modification. This is an epigenetic mark that we heard about yesterday in Molly's talk, but I'm not going to talk much further about that. I'll just be focusing on the transcription factors. So the way that we do this is we're looking for these SNPs or QTLs affecting transcription factor binding. So we call them binding QTLs or bQTLs. And the way that we visualize it is by taking each SNP and plotting its allele frequency before the ChIP on the x-axis and after the ChIP on the y-axis. And so the vast majority of SNPs, when we do this for one TF here, shown for NF kappa B, the vast majority are right on the diagonal, which is where you'd expect if they have no effect on binding. But we do see quite a number of SNPs falling off the diagonal as well. And those are these binding QTLs that we're interested in. So when we have a new method, it's a good idea to compare it to the standard method to see if we're actually in agreement. So there was one study that took 47 different individual LCLs and did ChIP-seq on each one of those. So this is the non-pooled approach that I referred to earlier. And they did this for one of the same TFs that we looked at called PU1. And so we can compare our results to theirs to see how well they agree. So first of all, when we just look at when we have a bQTL, how often do we call the same allele as being higher affinity as compared to this previous study? It's the vast majority of the time, 99%, we see the same allele as being called as higher affinity. And we can look in more detail even and ask when we estimate the quantitative effect sizes of these different bQTLs, then in our pooled approach on the x-axis, we see pretty good agreement with these individual level samples on the y-axis. So we're getting not just a directionality agreement, but also the quantitative effect sizes is also mostly in agreement. So we think that we're getting some pretty reliable results here with this pooled approach. Now we can also ask, comparing to the same study, what is the efficiency of this method? So if we look at QTLs per sequence read in our study, we see that we do about 25 times better than the non-pooled approach from this same paper. If we do it in terms of QTLs per ChIP performed, then it's about 75-fold better. So we're getting a pretty big increase in terms of the efficiency and a decrease in cost. Now, we can ask a number of questions now that we have this catalog of these bQTLs. So first of all, just what fraction of SNPs are affecting binding? So out of SNPs that are actually bound by these different transcription factors, between 1% and 3% of them typically show up as a significant bQTL. So it's a small percentage of SNPs, but there's a lot of SNPs that are bound. So this turns out to be quite a large number across the genome, anywhere from a few thousand up to 40,000 SNPs per factor are classified as these bQTLs. And so this is quite a large number, which allows us to ask some pretty interesting questions that we wouldn't be able to ask without this type of data set. So one thing we observed was that many of these bQTLs actually are significant for multiple factors. So we might see one SNP associated with the binding of multiple TFs, and then one question that we wanted to ask then was what is actually happening here in terms of the causality? So who is recruiting whom to these binding sites? Is it NF-kappa B recruiting JUN-D or vice versa? And we can actually figure that out by incorporating data from the known motifs for these different factors. So both of these TFs have known short DNA sequence motifs that they tend to bind to. And if we see that a SNP that's a bQTL is in an NF-kappa B motif, then we can be pretty sure that its direct effect is on the binding of NF-kappa B to the DNA, and any other effects on other TFs must then be indirect. And same thing we can do for JUN-D. And when we do that analysis, we see that both of these cases actually occur quite often, much more than you'd expect by chance. And so we can say that either one of these factors is able to recruit the other one to its binding sites. Now that we've abstracted this to the level of motifs, we don't have to restrict ourselves just to these factors that we've done the ChIP for. We could look at any motif that we can imagine and ask whether those motifs are enriched for bQTLs. So what I actually did was take a set of thousands of different human motifs and just ask which ones of these are enriched for bQTLs, and we see lots of enrichments. But the most interesting one that I want to mention is that we found just one motif that was enriched for the bQTLs of all 5 TFs. So it can recruit all 5 to its binding sites. And interestingly, that was a motif for a protein called CTCF, which is a very well-studied protein. It's involved in chromosomal looping. And this is interesting because it didn't actually was not previously known to have a role in recruiting all these different TFs to its binding sites. So that's an interesting finding that came out of the data. Now we can also look at long-range effects of these bQTLs across large swaths of the genome. So here's one example of a particular binding site for a TF called SP1. It recruits all 5 of our TFs to that binding site, and there's a bQTL there that changes the binding for all 5. When we look around there in the genomic region, we see that it actually regulates the expression levels of 5 genes nearby, so these 5 that are colored in green, including one that's over 500 kb away, so a pretty long-range effect. But interestingly, it does not affect the expression of one gene that's actually closer, called CALD1 here. And so we wondered, why would it affect the expression of all these various genes nearby, but selectively not for CALD1? And so to answer that, we looked at chromosomal interaction data that was previously published. This is known as Hi-C data. And this just gives you an idea of where different chromosomal regions are interacting with one another in the 3-dimensional space of the nucleus. And when we look at where our SNP falls and what it's interacting with, it interacts pretty highly with these various transcription start sites for these genes, but specifically does not have much of an interaction with CALD1. And so we think in this case, it's not regulating the expression of CALD1 simply because it's not nearby in the 3-dimensional space of the nucleus to this gene, and so it's not able to have an effect. And this is a pattern that we see recurring over and over again at different loci throughout the genome. So we can represent these interactions by these arrows, and so there's no arrow going over to CALD1. So now, Brian He, who's an undergraduate in the lab, decided to ask a separate but related question. So we can see that bQTLs have effects on genes determined by their long-range interactions, as I just showed you. But a separate question is whether bQTLs can actually affect the chromosomal interactions themselves. And this is something we really didn't know what to expect for, because CTCF, this protein that I just mentioned, has a very well-established role in determining which regions of the chromosome interact with each other. But none of these transcription factors had any known role in that process as well. So whether they're bQTLs, which are affecting their binding, would affect these chromosomal interactions, we really couldn't say. So Brian just looked at the data and asked, when we see these bQTLs for our 5 TFs, you have one allele that's higher affinity and one that's lower, and he asked, is there any difference in the extent of long-range interaction between these 2 alleles? And it turns out the answer is yes, and for all 5 transcription factors, we actually see the same pattern, and that pattern is that when you have less of the TF bound, then you have fewer long-range interactions. And this is a very strong result across all 5 of the TFs. So it appears that, that these bQTLs are actually contributing to the 3-dimensional architecture of the chromosomes within the nucleus. And so it's actually a much more complicated picture than just CTCF regulating this. There's a number of other transcription factors that are playing a role as well. All right, so now with all that, we did all that fun analysis, and then we could get back to the actual question that motivated this study, which is how are these genetic variants in the non-coding regions affecting these complex traits at the molecular level. And so we can go back to this Venn diagram and ask what is in here in this intersection. So I'll give you a few examples. We can start with some very simple examples where we just have a bQTL within a particular motif. This is for NF-kappa B, and it affects the expression of a nearby gene, which affects your risk of getting a heart attack. So there's very simple examples like that. We can look at some more complex examples. So here's one, a very well-known gene called cyclin D1, which is a cell cycle regulator, and it's regulated by two well-known enhancers located upstream of it. And interestingly, these two enhancers have genetic variants within them that specifically affect risk of getting just one type of cancer. So what we did was look in our bQTL catalog and actually found a third enhancer that's located much further upstream, over 400 kb away from the gene. And this has a bQTL in an NF-kappa B motif, and that specifically affects your risk of getting prostate cancer. So it's a third enhancer that had not been recognized before, and that also affects risk of getting cancer. A third example is with Crohn's disease and this gene called IRGM. This has been very well studied in the context of Crohn's disease, but there's actually two independent regions of association around IRGM. One of these regions has been very well studied, and there's been causal variants that have been found and lots of papers published on it. The other region has not received much attention at all because there's a big block of LD, so there's lots of SNPs that have equally strong associations, and they're all in this non-coding region, and nobody really knew what to make of it. So what we found was that one of these SNPs falls into a PU1 motif and has a very strong bQTL for PU1, but also has effects on JUD and NF-kappa B. And so we think that this PU1 is recruiting these two to its binding site and then affecting the expression of IRGM and thus risk of Crohn's disease. So these are just a few examples, but really this is just the tip of the iceberg. We have over 2,000 of these GWAS hits that are also bQTLs. And so while we can't say for sure without further experiments, these create some very clear and testable hypotheses for the molecular mechanisms of these GWAS hits. So to conclude, what we've seen here is that this pooled ChIP-seq method can very efficiently map QTLs for molecular level traits like TF binding and histone modification. These QTLs are natural genetic variants segregating within humans that affect TF binding and in many cases also affect disease risk. And the extent of this overlap between the GWAS hits and the bQTLs suggests that there really is a major role of TF binding in our risk of getting complex diseases. And this is something that we really haven't been able to make this conclusion before because nobody's really had a data set of all these bQTLs to compare to the GWAS and actually ask, is there much more overlap than expected by chance? So now we see that there is, which tells us that these TF binding is probably playing a major role in human phenotypic variation. And so this approach, we're pretty excited about it because it can be applied to many other types of traits and tissues, different populations and species. So to give you one example of this, so Irene Kaplow, who's a graduate student and also a SEG fellow, has applied a similar type of approach to DNA methylation. So DNA methylation is another epigenetic mark that can affect transcriptional regulation. And so she applied bisulfite sequencing to pooled human samples and was able to map over 2,000 QTLs affecting DNA methylation, once again, including a number of these GWAS hits. So the next steps that we're excited to take here is to expand this beyond the one population that we've started with. So both of these studies in methylation, as well as TF binding, have been focusing on this Yoruban population from Nigeria. But as you know, humans are very diverse, and there's a lot of interesting biology that we can learn from studying different populations. And so we know from previous work in our lab, as well as others, that DNA methylation differs between populations. But what we don't know is whether these bQTLs may differ as well, and whether these may underlie population-specific traits, because if they're contributing so much to complex disease variation, maybe they could also contribute to population-specific disease risk, which is known to be pretty common. So what we're doing now is mapping QTLs for chromatin accessibility using a ChIP-seq, and doing this in 1,000 different individuals from 10 populations. And this is really exciting because doing this in 1,000 individuals is something you couldn't even fathom with a single unpooled individual level approach, but with the pooling it becomes quite accessible. All right, so with that, I would just like to acknowledge Ashley, who did the vast majority of this work, as well as Irene and Brian, and some other contributors as well. And I'd like to thank my lab for being awesome, and our funding as well. And thank you for listening.
Awesome, looks like we already have a question in the back there.
First of all, I'd like to say happy birthday. Happy birthday, Gunther.
Thank
Happy
you.
birthday.
You're supposed to sing that, by the way.
Yeah, I was thinking about singing it, but then I thought we'd waste time for questions. So I've asked you this before, and I'll ask you again now that you have a lot of data on it. But you know, one of the flaws with doing it this way is that you lose mutations that are rare. You know, SNPs that are rare and affect binding in a negative manner or become very difficult to detect in the pooled data. Do you have a sense for how your what your range, what your limits are, what your detection ability is, and how much some sense of how much you're missing because of the pooled strategy? So you talked about things you found in common, but not things that you missed that maybe some other strategy would find. Right.
Yeah, that's a great point. So I think we are definitely missing quite a bit there. So just to explain to everyone else, what Mike is referring to is if you imagine that pre-ChIP versus post-ChIP plot, if you start out, let's say at a very low allele frequency, there's a lot of room to go up, but not much room to go down. And so there's an asymmetry there, and so you can't do much, you don't have much power to detect those effects that would be pushing you down further than you started. So I think the solution to that, in some respects, is bigger pools and more sequencing, because if you have a big enough pool, like of 1,000 individuals, a rare variant might actually be common enough at that point that you can actually see these effects going down. But there's always going to be some asymmetry. So we don't have a good idea of how much we're missing, because it's quite possible that those rare variants are actually enriched for functional variants, and so we can't really extrapolate from the rest of the range. So I'll have to say I don't know how much we're missing. We're probably missing quite a bit. And the most we can do with this approach is try to scale it up and see how much more we can get from bigger pools and more sequencing, and that'll start to answer it. But yeah, there'll always be some asymmetry inherent in that.
Another question? Right here in the middle. I think closer, maybe a little closer to you.
Hi. So I was interested in the shared bQTLs for the different TFs. Do you know how how that sharing is being mediated? Does it involve like external chromatin looping, or is it just like kind of a local interaction right at the binding site?
We don't know. That's a great question. So how are these indirect effects actually happening? So in the case of CTCF, our best guess, and this is just a total guess, but we don't think it's likely that it's physically interacting with all these different TFs, just because that's something that has never been seen for CTCF. So we think in that case, given the known role of CTCF in looping, it's quite possible that it's just making the local chromatin more accessible and maybe looping in other regions of the genome that have these TFs bound to them, and then we're able to see them as binding to these CTCF sites more often when CTCF is present. So for CTCF, I think that might be a likely explanation, is that looping is important. For the other cases, like the one I showed with NF-kappa B and Jun-D, we really don't know. In that case, it could be more of a local physical interaction-based effect, or the alternative might be that there could just be one of them binds to the genome and remodels some nucleosomes, making another site more accessible right next door, and then that allows another factor to come in, even without physical interaction between them. So our data don't let us distinguish between those.
One more question?
So obviously one challenge is that you're limited in how far away from where the actual binding is happening you can look for variants that are affecting binding. So is that affecting your choices on like what sequencing length you're using? And also I'm interested how you think that's going to play out with something like attack-seq, because I would imagine with the TF binding, you're expecting very local effects, whereas with the attack-seq, if it's something that's, you could imagine having variants further away, which are going to affect binding of something there, and that's going to have knock-on effects somewhere else that you're not going to be able to pick up as easily.
Right, yep. So yeah, so our method is restricted to whatever is covered by the sequence reads, and so we do try to get as long reads as we can and do paired end, so we get as much information as possible there. But yeah, that's definitely a limitation. And like you said, for the TFs, there is evidence that the vast majority of information that determines TF binding is very local. So you can take 80 base pairs around a binding site, put it into a plasmid, and that will almost always recapitulate the level of binding you see. So we think for TFs, it is highly local, and we're probably capturing most of that with the sequence reads. For the attack-seq, we're not entirely sure how much of that sequence variation we'll be capturing. We think it will be still the majority because studies like of DNase hypersensitivity QTLs from Jonathan Pritchard's lab have shown that even for that, it is still very highly local enrichment that you see right in the DNase hypersensitive peaks for these ds-QTLs. And so the attack-seq we expect will be pretty similar because it's also looking at chromatin accessibility. But for sure, there will always be some that are further away that we won't be able to capture.